वीडियो तो चल नहीं रही होगी।
अब सर यूट्यूब पे जाइए सर, ऊपर देखिए, इसे ऊपर करिए, बैक मत कर दीजिएगा, आपका शुरू हो गया सर।
इसको हम शेयर नहीं कर सकते सर?
अब सर शेयर करने के लिए ये जो एरो बना हुआ है ऊपर, ये वाला
नहीं मतलब
कॉपी,
स्क्रीन शेयर।
स्क्रीन शेयर हो रही है सर आपकी, इसको लिंक को कॉपी कर लीजिए यहाँ से, वीडियो लिंक को ना, राइट हैंड साइड से सर, ऊपर ऊपर इससे, हाँ इसे बस क्लिक करें, कॉपी हो गया सर।
नहीं, स्क्रीन शेयर कैसे होगी सर?
सर स्क्रीन शेयर हो रही है सर अपने आप ही, आप देखिए सर यहाँ पर
ओके
लाइव
ओके
स्ट्रीमिंग
ओके
चल रही है,
ओके ओके ओके।
तो सब देख रहे हैं सर।
तो मैं
इसे
सेंड कर देता
क्रॉस
हूँ।
कर दीजिए इसे, बस ये इसको क्रॉस मत करना सर, सारी स्ट्रीमिंग क्रॉस हो जाएगी। टेलीग्राम पे सर,
टेलीग्राम कर लेता हूँ मैं।
बस यूट्यूब खोलिए अपना, क्रोम, तो यहाँ पे सर लिख दीजिए गुड इवनिंग क्लास, 14 बच्चे आगे सर होंगे। सर आप एक्चुअली सुन नहीं सकते यूट्यूब स्ट्रीमिंग में, यू कैन ओनली पार्टिसिपेट विद दी स्टूडेंट्स बाय चैटिंग। Confirm initially with the students, is my screen being shared, and is my voice clear enough to all of you?
हेलो हेलो पार्टिसिपेंट्स, हेलो पार्टिसिपेंट्स।
Streaming, यहाँ पे रिप्लाई करेंगे बच्चे।
ओके ओके क्लियर। ओके थैंक यू।
Yes, live streaming को बोलिए सर। आज इंट्रोडक्शन दीजिए अपनी।
Good evening to all. Navdeep Gupta, good evening. Diksha, Sachin Saurabh, good evening. Shreya, hello. So we are here to learn concepts of bioinformatics, right? Just after 4 to 5 minutes, we will start. Please confirm me which language you will prefer, Hindi or English, so that I will proceed in that language. You are more comfortable in Hindi or English? As I am seeing your comments, uh, participants commenting on both language more, right? So we will discuss here in both language, Hindi and English simultaneously. Okay, no problem. If you have any problem, then I will solve it. Okay, thank you. We should wait for 1 to 3 minutes more to join other participants. 72 participants have been joined, but we are waiting for others because in this course, 130 approximate participants will take part. Okay. So this course, those session will be very much interactive because I am not going to only speak. You will also comment whatever you are learning. You will uh, give me a comment. Okay, you are understanding or not. Then I will decide. I am teaching you or you are getting anything concerned with the bioinformatics that is helpful or not. Okay, so first of all I would like to introduce myself. I am Dr. Anand Kumar. I have completed my PhD from IIT BHU in vaccinology, right? And I am working as an assistant professor of biotechnology. I have more than 6 to 7 years of teaching and research experience. I have a postdoctoral experience in the vaccinology and transgenic cell line development and virology, vaccine development, right? So you all are a student of biotechnology, right? Or other streams of life science. Please comment in which stream you belong. Okay, so I can understand in which way I should discuss the bioinformatics, biotechnology, biotechnology. Good. Please comment. Good. Most of the pupils are from biotechnology and biomedical. Good. Zoology honors, animal biotech, genetics. Very good. Biotechnology, microbiology. Very good. Toxicology, MBBS. Very good. Botany, BTech biotech. Okay, second year. Zoology, chemical engineering. Good. So you people belong to biotechnology and different fields of life science like bioinformatics, zoology, plant biotech, animal biotech, and genetics, right? Even you belong to pharmacy background. Good. So what what is the need of bioinformatics? Why we are discussing here bioinformatics? What is the role of bioinformatics? We will discuss here, right? So you are human or living being. you have DNA, right? Everybody knows DNA. But DNA is made up of ATGC, and in the world only ATGC is present in the DNA. No other nucleotide is present, right? So sequence of DNA and number of nucleotide, it will depend upon the diversity we have, right? So we will discuss here different topics today. Introduction of bioinformatics and historical overview of bioinformatics, human genome project, and biological databases, internet and bioinformatics, knowledge discovery and data mining, career prospect in bioinformatics. Okay, so living being. If we talk about the living being have a DNA, and in human, this DNA is a very long, approximately 3 billion base pairs it contains, right? मीन्स आपका जो डीएनए है वो कितना बड़ा है? 3 बिलियन. 3 बिलियन का मतलब आप समझ रहे होंगे, मीन्स 3000 मिलियन बेस पेयर. आप एज्यूम कर सकते हैं कि ये इतना बड़ा डेटा आप कंपाइल नहीं कर सकते हो, राइट? मीन्स इसको आपको एनालिसिस करने के लिए बहुत बड़ी प्रॉब्लम सफ़र करनी पड़ेगी, राइट? तो इसके लिए आपको क्या करना है? इसको आपको एक डेटाबेस तैयार करना पड़ेगा, राइट? तो डेटाबेस तैयार करने के लिए आपको एक बड़ा प्लेटफॉर्म चाहिए, ठीक है? एक तो आप कॉपी पर नोट कर सकते हो, राइट? देन आपको क्या करना पड़ेगा? ये जो एटीजीसी का जो सीक्वेंस है, बड़ा होने की वजह से इसको आप लोग दूसरी चीज़ क्या कर सकते हो? अपने सिस्टम में कंप्यूटर की हेल्प से उसमें भी फीड कर सकते हो, राइट? तो जो बायो इन्फॉर्मेटिक्स को हम लोग समझने की कोशिश करते हैं, बायो इन्फॉर्मेटिक्स जो टर्म है वो डिसाइड करता है कि इसमें बायोलॉजिकल इंफॉर्मेशंस को मैथमेटिकल और कंप्यूटर की हेल्प से कंजर्व किया जाता है या फिर एक जगह स्टोर किया जाता है, राइट? तो हम लोग जो अभी स्टडी करने जा रहे हैं, तो कंप्यूटर साइंस, मैथमेटिक्स एंड बायोलॉजिकल साइंस, तीनों का जो मिला के हम लोग बनाते हैं, तो इसको हम बोलते हैं बायो इन्फॉर्मेटिक्स, राइट? तो इसको हम लोग डेफिनेशन जानने की कोशिश करते हैं इसकी. A computational approach to solve the problem of biological data, right? There is some problem, technical problem, right? So now bioinformatics is emerging and advanced branch of biological science, contain biology, mathematics and computer science, right? Everybody knows bioinformatics developed a new thought to maintain the concepts and store the huge amount of biological data, right? Bioinformatics concepts and method are different than the biological concept and method, right? Bioinformatics a logical and technical means by which not only solve the biological problems but also can predict the new aspects, right? We belong to biological science, right? So here we will focus more on the genomics and proteomics study, because in the biotechnology or zoology, in microbiology or in other fields, we will study only for the genomics and proteomics. Genomics means study of genetic material and proteomics means study of proteins. More than thousands proteins are present in a living organism, and that all the proteins developed from the genetic material. Whatever the genetic material we have, this will decided uh, uh, this will decided by the our inheritance. Our our parents are the sorry it will decided on the our forefather or parents, right? तो हम लोग क्या कर सकते हैं, इसमें जो बायोइन्फॉर्मेटिक्स है, बायोइन्फॉर्मेटिक्स में हम लोग जीनोमिक्स और प्रोटियोमिक्स को सिमल्टेनियसली स्टडी करेंगे। मीन्स हमारा जो जेनेटिक मटेरियल है, वो जेनेटिक मटेरियल ही हमारा डिसाइड करेगा हमारा जो प्रोटीन्स हमारी बॉडी में प्रेजेंट हैं और जो कैरेक्टरिस्टिक फीचर्स हैं हमारी हमारे पास 2 हैंड्स हैं, 2 लेग्स हैं, ठीक है, 2 ईयर्स हैं, 2 आइज हैं, ये डिसाइड कौन कर रहा है, हमारा जेनेटिक मटेरियल, राइट। तो इसके जो बायोइन्फॉर्मेटिक्स है, बायोइन्फॉर्मेटिक्स में हम लोग अभी जो डिस्कस करेंगे वो जीनोमिक्स और प्रोटियोमिक्स पर फोकस करेंगे। बट जो बायोइन्फॉर्मेटिक्स है वो अदर फील्ड्स को भी कवर करती है, लाइक कंप्यूटेशनल बायोलॉजी, डेटाबेस मैनेजमेंट सिस्टम, सिस्टमैटिक बायोलॉजी, कीमो इन्फॉर्मेटिक्स, बायोस्टैटिस्टिक्स और कंप्यूटेशनल लैंग्वेजेस लाइक सी, C++, पर्ल, बायोपर्ल, बायोजावा, राइट। तो हम लोग बायोइन्फॉर्मेटिक्स के अंदर सबकी स्टडी करते हैं, बट हम लोग ये जो कोर्स है इसमें हम लोग फोकस करेंगे जीनोमिक्स एंड प्रोटियोमिक्स। आप लोगों ने मॉलिक्यूलर बायोलॉजी की स्टडी की होगी, जेनेटिक इंजीनियरिंग की स्टडी की होगी, आप लोग PCR को भी अच्छी तरह से जानते होंगे, ठीक है। तो जो वेक्टर होता है, जो वेक्टर होता है, वेक्टर को कैसे हम लोग डिजाइन करते हैं, इसमें प्राइमर कैसे हम लोग डिजाइन करते हैं, ये सारी की सारी चीजें हम लोग स्टडी करेंगे। हम लोग 3rd या 4th लेक्चर में प्राइमर डिजाइनिंग सीखेंगे। बाकी जितने भी डेटाबेस हैं अभी तक उनको हम लोग एनालिसिस करेंगे। होमोलॉजी मॉडलिंग क्या है, प्रोटीन मॉडलिंग क्या है, ठीक है। अब सीक्वेंस कैसे सर्च करते हैं, प्रोटीन सीक्वेंस है या फिर न्यूक्लिक एसिड सीक्वेंस है, इसको हम लोग कैसे सर्च करते हैं, सारा का सारा हम लोग लर्न करेंगे। हम लोग स्टडी नहीं करेंगे, इसको हम लोग लर्न करेंगे, राइट। मैं आपको बताऊंगा इस नेक्स्ट सेशन में कि हम लोग जो प्राइमर डिजाइन करते हैं उसके लिए कुछ सॉफ्टवेयर्स होते हैं। मैं जो यूज करता हूँ वो जीन2 लाइट सॉफ्टवेयर है एंड DNA स्टार। आपको अगर कहीं से फ्री वर्जन अवेलेबल होता है देन प्लीज डाउनलोड इट एंड इंस्टॉल इन योर कंप्यूटर। दैट विल हेल्प यू टू लर्न more appropriately for the practice, right? So today I will give you just an overview of bioinformatics, but the next session will be totally practical based, right? How you will predict a protein on the basis of homology, right? How you will prepare a ORF, open reading frame, for the expression of any protein, right? Okay, someone are saying, please repeat. Some technical problem is there, right? Maybe they have joined late. So I will repeat the things which are more important. No problem. Okay. So I am sharing you PPT so we can discuss the overview of bioinformatics, and I will definitely share with you that PPT. Okay, so you will get it basic idea about the bioinformatics, right? You can note gene tool light. I will comment you gene tool light, and other one is a DNA star. So you can download it if a free version is available. Okay, so that will be very beneficial for you, because you will learn how to design primer. Okay, generally people do not design because they don't know how to design primers, right? They only use already given primers in our previous published research papers. Okay, but I will tell you, and you will learn, definitely you will learn how to design primers and how to maintain reading frame for the expression of any gene or protein, right? So here I am going to discuss the next thing. Now, in the computational biology, this is also field of bioinformatics, that is also known as in silico biology. Okay, in the computational biology, we will study for the comparative homology modeling. We will learn how we will perform homology modeling. We will perform through a Swiss model. Okay, in our next class. Next is phylogenetic analysis. We will check the evolutionary relationship with other organism for one organism through phylogenetic analysis. Okay. Next is protein structure prediction. In the protein structure prediction, we will use different tools. I will show you how to predict protein structure, because different types of structures are there: primary, secondary, tertiary, and quaternary, right? But uh, if you want to know the structure of protein, then you will perform X-ray chromatography and X-ray diffraction and NMR, nuclear magnetic resonance. Okay, then you will know what is the structure of this specific protein. Next is the protein folding problem. Okay, we will study in detail this also. Next is microarray. Everyone knows what is microarray. Microarray will be used for the detection of any gene, protein, or anything. Okay, in the microarray, lots of samples we can check in a single time. Okay, next stage docking approaches and new drug discovery. If you want to discover a new drug, so you will perform a docking study. Okay, ligand receptor binding approach. So we will study this in a computational biology one by one. Now we will discuss here the basics of the history of bioinformatics uh, related with the molecular biology. Okay, means related with the DNA and proteins discovery. Okay, in 1859, the on the origin of species, that is the book published by Charles Darwin. Everybody knows the name of Charles Darwin, that introduced the theory of genetic evolution, because everybody knows about the evolution. What is evolution? Okay, how many people you know about the evolution? Okay, someone saying that it will be very good if you will take uh, class on the Zoom. I think participants are more, and Zoom will allow only for 100 participants. Okay, therefore that will be difficult. Okay, most of the pupils know about the evolution. Okay, good, very good. Before coming to next point, please subscribe my channel first. Okay, please, all participants, please subscribe my channel. Please subscribe my channel. Okay, good. Keep on. So we will back. So we were discussing about the origin of species, that is the book published by Charles Darwin, that introduced the theory of genetic evolution. Most of the people commented, we know about the evolution. Okay, allow adaptation over time to produce organism best suited to the environment. Okay, next is in the 1869, Frederick Mischer, he was the scientist, firstly isolated nuclei from the blood cells. Okay, then from the nuclei he isolated DNA, and he gave the term nuclein. Okay, so DNA comes under the nucleic acid. Nucleic acid means DNA and RNA, right? So you have to remember this thing. Nuclein term given by the Frederick Mischer, okay, in 1869. Next point is in 1951, Linus Pauling and Corey proposed the structure for the alpha helix and beta sheet. You know, in the protein, alpha helix and beta sheets remain present. You know, phi psi angle, Ramchandran plot, you have read. Okay, I know this thing. How many people know about the Ramchandran plot, phi psi angle, and alpha helix and beta sheet? I think everybody knows this. Okay, okay, most of the people knows. I think it will be more clear to all. Okay. In 1953, Watson and Crick proposed the double helix model for DNA based on X-ray data obtained by Franklin and Wilkins. Because Franklin and Wilkins uh, knows more about the X-ray. Okay, that's why he gave the structure of a DNA double helix. Okay. So after that, in 1955, the sequence of the first protein to be analyzed was bovine insulin. That was announced by the Frederick Sanger. You know about the Sanger gave the who gave uh, the techniques of DNA sequencing. We call it Sanger sequencing method. Okay, everybody knows this. So you have to remember this thing. This is a very important point. First protein that was sequenced is a insulin, bovine insulin. You have to remember. Okay, I will ask in the next session. Okay. Next point is 1958. The Advanced Research Project Agency is formed in the US. Okay. Now we will talk about the history and scopes of bioinformatics. In 1973, the Brookhaven Protein Data Bank is announced. Okay. PDB. PDB means Protein Data Bank, that was established. In 1987, Pearl is released by Larry Wall. Okay, Pearl. What is the full form of Pearl? Practical Extraction Report Language. Okay, you know about the C, C++, Java, Pearl. Okay, so this is the language. Practical extraction report language. This is also important for you. You have to remember this thing. Next point that is also important, in 1988, you have to remember the year. Okay, 1988, National Center for Biotechnology Information, that is in US, founded by NIH, National Institute of Health. Okay, this is a big institute governed by the US government. Okay, after that, in 1990, Human Genome Project was launched. Okay, Human Genome Project means Human Genome Sequencing Project. Okay, everybody knows about the Human Genome Sequence Project. So in this BLAST program introduced by S. Carlin and S.F. Edsel. Okay, so you have to remember the name of scientist S. Carlin and S.F. Edsel. Next is Tim Berners-Lee, a British scientist, invented the World Wide Web, means WWW, in 1990. Both are important. Name of scientist and year. Okay, everybody knows WWW, World Wide Web. Okay, so this was invented in 1990. But in 1992, the Institute for Genome Research, associated with plans to exploit sequencing, okay, commercially through gene identification and drug discovery, was formed. Okay, next is in 2001, first genome, means human genome, was published, that is of 3 billion base pair, or you may say it 3000 million base pair. Okay, you have a genome size of 3 million, sorry, 3 billion base pair or 3000 million base pair. You can assume it will be how long? Can you assume 3 billion base pair? Okay, I will explain Sanger sequencing, okay, later. First, we will finish this thing, background of bioinformatics. Then I will definitely tell you what is Sanger sequencing. Okay, this is a basic method of sequencing. Sequencing means first you can say that sequence of ATGC, right? So there are different methods like Maxim Gilbert sequencing and the Sanger sequencing method, automated method. Now, right now, the most common method is next generation sequencing and latest one. Okay, so I will tell you what is Sanger sequencing. Okay, so in the human, can you assume more than 1 lakh genes are present in your side, in your DNA? More than 1 lakh genes are present. Can you assume the size of your DNA? That is very large. If you will collect your DNA, then your DNA will reach to the sun. This is not a simple thing. This is a very vast. Can you imagine? So okay, uh, I will try. Okay, okay, I will try. Okay, definitely I will cover. Okay, next one is in 2010 completion of the 2010 project to understand the function of all genes within their cellular organism and evolutionary context of Arabidopsis thaliana, right? We hope in the 2015 to complete of the first computational model of the complete cell, or maybe even already of a complete organism. Okay, we expect this. uh, one thing here I would like to discuss, that you think that 1 lakh, more than 1 lakh genes you have. Okay, we have to identify which gene expresses specific proteins. You have, if you have 1 lakh genes, then more than 1 lakh proteins or less than 1 lakh protein you may have. Okay, and all the proteins have different functions, and you are very much complex organism, right? So your complexity is directly proportional to your genetic material. Your genetic material is more complex, then you will be more complex. Uh, just imagine that a bacteria have a very simple genetic material, and the size of bacterial genetic material is very less in comparison to human genetic material or genome. Okay, can you imagine that plant genetic material or genome have more or bigger than human genome? Means human is very much simple than wait a minute. Okay, I will share you. Means I am trying to say that bigger than human genome, means the complexity of plant is more, means plant is more complex than human. But you are thinking that I am more complex, but you are not more complex, plant is more complex. Okay, I am not saying this. Who is saying? This is saying genetic material, the analysis of genetic material. Okay, now we will talk about the human genome project. This is the US government project coordinated by the Department of Energy and National Institute of Health, launched in 1986 by Charles Delici. Okay, first we will try to know what is genome. Okay, uh, in a single cell, haploid number of chromosome, we will call it genome, right? Haploid number of chromosome in a single cell, we will call it genome. Okay, we can say it the whole hereditary information of an organism that is encoded in the DNA. This is the another definition of genome. You may write here. Now we will discuss about the aim of the project. The aim was to identify the approximately 1 lakh genes in the human DNA. Means we know after sequencing, we know that genome size is 3 billion base pair, and genome have more than 1 lakh or 1 lakh genes. But what is the function? What is the role? What is the characteristic feature of specific gene and all genes? We have to know. That was uh, uh, that was the research in a human genome project, and aim was also. Next was the to determine the sequences of the 3 billion bases that make up human DNA. Okay. Next was the store this information in database. Can you assume that 3 billion bases data can you store in a copy or book or anywhere? That is a very big data. Okay, after compiling this data, you have to analyze this data. That is a very big task. Okay, so therefore you have to use bioinformatics tool. Without bioinformatics tool, you cannot perform this task, right? After that, store this information in databases. For the nucleic acid, you can use different databases like NCBI and PDB. These are the most common databases, and DDBJ, DNA Data Bank of Japan, that that was uh, constituted by the government of Japan, and NCBI constituted by the government of USA. Okay. Now address the ethical, legal, and social issues that arise from genome research. Okay, that is a very much important point. You have to know ethical issues. Okay, if you are using genetic material of human, uh, some governments uh, uh, some governments are very concerned about this. Okay, they do not allow if you are using genetic material of human. Okay, now we will talk about the basics of sequencing results, or history of sequencing. The first organism, that is bacteria Haemophilus influenza, okay, that was sequenced, complete genome was sequenced of Haemophilus influenza. You have to remember the name of bacteria Haemophilus influenza, that was the organism firstly sequenced. Okay. Next point is the approach called whole genome shotgun sequence, to sequence the 1749 genes of the bacterium in a minimum time period. Okay. Next is the Haemophilus influenza project was based on an approach to genomic analysis using sequencing and assembly of unselected pieces of DNA from the whole chromosome. Okay, you can see here the mitochondrial DNA of human. Okay, the mitochondrial human DNA have a 16,569 base pairs. Okay, you can see here, this is the mitochondrial DNA of human. So in this slide you have to remember the name of bacterium that was firstly sequenced using genome shotgun sequencing. Okay. Now we will discuss here benefits of human genome project research. What were the benefits? Improvements in medicine and drugs used in genetic or metabolic disorder. Okay, you know about the genetic and metabolic disorder very well. Disorder comes from the mutation in genetic material or in metabolic pathway, that is known as genetic or metabolic disorder. Next is a microbial genome research for biofuel and environmental cleanup. You know, superbug or lots of microbes which clean up the oil spilling. Okay, so we can create a microbe that can clean the oil spill. Okay, next is DNA fingerprinting and forensics. You know, DNA fingerprinting, that is mostly used for the forensics, to know identical twins, or to know the suspected persons or criminals. Okay. Now next is improved agriculture by improving the bite gene of high yielding variety of grain and livestock. Okay, so this is the important thing, better understanding of species evolution and human genome, And next is more accurate risk assessment by gene mapping. Okay, uh, you know, the genetic material, only the game of ATGC. We are changing the sequence and number of ATGC. We are uh, we are so much diverse because of ATGC. Now, this is the basics of internet and bioinformatics, how we can correlate. You can see here, we will not more focus. Internet plays an important role, everybody knows, to retrieve the biological information. Bioinformatics, emerging new dimensions of biological science include the computer science, mathematics, and life science. Everybody knows, uh, not a new thing. The computational part of bioinformatics used to optimize the biological problems like metabolic disorder, genetic disorder. Computational part contains software and tools development, operating system like Win 2000, XP, Linux, Unix. These are the operating systems, and database development, and software and tool application. This is the basics of computer science we will use. Now the internet and bioinformatics, the mathematical portion helps to understand the algorithm used in bioinformatics software and tools. Now the mathematical portion which used in in bioinformatics are matrices. This is the mathematical portion, you know, MSA and phylogenetic prediction, BLAST, FASTA, and biostatistic, HMM, ANN in secondary structure, artificial neural networking. ANN means artificial neural networking in secondary structure prediction. Now next point is complex mathematical functions, Fourier transformation. Next differentiation, integration, time space complexity, E value, P value, and BLAST. So we will not be more focus on these things. This is a mathematical approach, so we have to know just the name and the overview. Okay, now here the internet is a global data communication system. It is hardware and software infrastructure that provides connectivity between computers. Now, the web is one of the services communicated by the internet. It is a collection of interconnected documents and other resources linked by hyperlink and URL. Okay, so in manufacturing information, employee ID, supply, inventory, customer record, and product data. This is just this is only for the learning the concept of